In this video, we analyze our 16S PCRs on an agarose gel. First, let's have a quick look at antibiotic production by our soil isolates. Some of our soil isolates grown on SFM are inhibiting the growth of M. luteus. While all 4 soil isolates grown on MYM inhibit the growth of M. luteus. The results are slightly different when the gram-negative E. coli DS941 is used as indicator bacteria. To identify our soil bacteria, we have carried out a PCR using primers that amplify the 16S ribosomal RNA genes. While the PCR is running, let's prepare an agarose gel. We mix 1 gram of agarose powder with 100 milliliters of TAE buffer. Remembering to loosen the lid first, we melt the agarose in the microwave. When our agarose is completely melted, we cool it to 55 degrees in a water bath, and then we are ready to pour our gel. We insert combs into the gel to form the wells, and then we pour the molten agarose into the gel former. Once the gel is set, we take it out of the former and rotate by 90 degrees so that it is ready to run. We then pour TAE running buffer into the gel tank to cover the gel. Our PCR has now completed 30 cycles. We will remove 5 microliters from each sample to analyze on our agarose gel. We label a tube for each sample and place 5 microliters of gel loading buffer into each. We take a 5 microliter sample of each of our PCRs and mix with the gel loading buffer. The gel loading buffer contains phycol to make the samples sink into the wells, SDS and EDTA to produce sharp bands on the gel and stop any enzymatic reactions, and colored tracking dyes so that we can follow the progress of our gel. When all of our samples are finally ready, we can proceed to load our gel. We remove both combs and load all of the samples, 10 microliters of each, consisting of 5 microliters of loading dye and 5 microliters of PCR reaction. The top gel contains PCRs using primers 27F and 519R. The bottom gel contains PCRs that used primers 27F and 1492R. Once all of the samples are loaded, we can place the lid on the gel and connect the lead to the power pack. We run this gel at 60 volts for about 1.5 hours. Check that the dyes start to move in the correct direction and look for bubbles of hydrogen and oxygen coming out at the plus and minus electrodes. It's a good idea to keep your labeled tubes as a record of your gel loading order. The pink dye runs at the position of approximately 500 base pairs. You can run this to near the bottom of each gel. We stain our gel in 0.5 micrograms per mil ethidium bromide, made up in TAE running buffer. Take care, ethidium bromide is a mutagen. In the teaching lab, we will use gel red stain, an alternative to ethidium bromide, which may be safer. The gel is stained in ethidium bromide for about 30 minutes with gentle shaking. After a quick de-stain in distilled water, the gel is moved onto a UV transilluminator and photographed with an orange filter. We center the gel in the field of vision . Zoom in so that the gel fills the frame, and then capture the image. A copy of this image is available for students on Moodle. We can now pick samples to sequence, remove primers and nucleotides with a PCR cleanup kit, and send for sequencing using the 27F primer.